that brings it in order.
Correct. Correct. Okay, so we're going to talk about an organelle now that we've already talked about a number of times, and that is the ribosome. Ribosomes are interesting because they have a unique construction, right? It's not a part of the endomembrane system. It's a membraneless organelle. It consists of just RNA and structural proteins. Okay, so
I have a question.
yeah, fire away.
Not that subject, but people with the Tay-Sachs disease,
Uh-huh.
are they more unstable than the other Jews? Like, within the civilization? Like,
So-
can you differentiate between the two, or is it just something internal?
Good question. So if you have Tay-Sachs disease, you usually die within a year.
Oh, so you don't have a chance to grow full...
Correct.
full-grown- okay.
Yep.
That answers my question.
Yeah, it's a recessive disorder, and that'll make more sense when we talk about genetics. So it's possible for individuals to carry that disorder without actually exhibiting that disorder. Yeah. If you lose the genetic lottery, then you get 2 copies of it, and then you exhibit it.
Okay.
Okay, so the ribosome. Is everybody okay if I erase this list so that I have a little bit more space to write?
Mm-hmm.
Maybe we'll take a picture. Okay. Okay, so our ribosomes their structure is they consist of RNA and proteins. And they're organized into 2 functional units. There is a large subunit and a small subunit. And it looks something like this. It kind of looks like a hamburger bun, really. Like that. So this is the large, and then this is the small. So the ribosome, its function, right, is to construct proteins. So the function is to construct proteins. And the way that it does that is it reads a chemical message from the nucleus. Remember, we don't want to send our DNA out of the nucleus. That defeats the purpose of the nucleus. The nucleus is supposed to protect our DNA. So we keep it compartmentalized. So what needs to happen is we need to make a copy of our DNA, send it outside, and then that copy of our DNA is read by the ribosome, and a protein is made from it. And so some copy of our DNA, which also happens to be RNA, comes along, and it's read kind of like, do you guys know what cassette tapes are?
Mm-hmm.
It's read like a cassette tape. So cassette tapes consist of, sad that I have to ask that question. It consists of, right, like a plastic case that has some plastic tape inside. And then what happens when you read a cassette tape is there's this little magnetic device inside your stereo that feeds that tape through it and reads the magnetic signature on that tape. So that's what's happening right here. We have an RNA copy of our DNA that's going to be read through in between the large and the small subunits like this. So this is an RNA copy of DNA for protein construction. was a time when we could just say, it works like a cassette tape, without having to say, do you guys know what cassette tapes are? That was a long time ago. Okay. So those are our ribosomes. Now, ribosomes, you'll recall, consist of RNA, and that RNA is manufactured in a portion of the nucleus called the nucleolus. And it's a highly condensed region of chromatin inside the nucleus. So when you look at nuclei, you're going to see kind of like a distribution of red stuff inside the nucleus, and then there's going to be a spot that's really, really dark. That's the nucleolus, and that's where ribosomal RNA is manufactured. Now, I mentioned last time that ribosomes can exist in two spots. They can exist either embedded in the endoplasmic reticulum, or they can exist floating free in the cytosol. The location of the ribosome determines the location of the protein that it's making. If the ribosome is located in the cytosol, that protein is going to stay within the cell. If the ribosome is embedded in the endoplasmic reticulum, generally, that protein is going to be shipped outside the cell. Okay, does that make sense? Okay with that? Okay, we're going to end up talking about mitochondria a lot more when we talk about cellular respiration, but this is just going to be a really brief introduction, okay? So, the mitochondria mitochondria is plural, mitochondrion is singular. The mitochondria are the power plants of the cell. So their function is to convert one type of energy into another. So I'll write function first this time. What I'm going to say is they convert chemical energy into ATP. Now, ATP is a really, really important molecule for us because that's cellular energy. When our cell does anything, when our body does anything, it's doing it with ATP. So the reason that we eat stuff is so that we can make ATP. And the stuff that we eat is either protein or fat or sugar, right? And mitochondria are able to convert proteins or fats or sugars into ATP. That's their job. Okay, so their structure. So like the nucleus, they consist of two plasma membranes. But here, the inside plasma membrane is way bigger than the outside plasma membrane. So it consists of two layers of plasma membrane. And the inner membrane is larger than the outer membrane. So what that means is mitochondria look something like this, and we always draw, for whatever reason, mitochondria in red. But they have kind of a smooth outer membrane that looks much like an overinflated football. It's kind of oblong like that. Super smooth. But the inner membrane, because it's so much larger than the outer membrane, super wavy like this. Yep. Importantly though, it creates two compartments. It creates an inner compartment and an outer compartment. So question, why is it that we have a huge membrane inside and a smaller membrane outside? Or asked differently, why is this so wavy inside? What does that increase?
The faster the protein is.
I liked the way you're thinking about it. It doesn't really expand and shrink very much, no, but that's a good thought. So the answer starts with surface, ends with area. Yeah, it increases surface area. And surface area is important, right? Because what that does is it increases the amount of space that we can have proteins embedded. So think about it, right? The mitochondria, its job is to take one form of chemical and convert it into another form of chemical. That's going to require a whole bunch of chemical reactions. And what is it that catalyzes chemical reactions? It's enzymes. And so all along this inner membrane, we have enzymes. And these enzymes are taking molecules and shifting them all around. And the more surface area we have, the more enzymes we can accommodate. So that highly convoluted, super wavy surface means more enzymes and more reactions and a more efficient mitochondria. Okay, does that make sense? Okay, each one of these projections, these finger-like things, are called cristae. So cristae is plural, crista is singular. Alrighty. So the next organelle we're going to talk about is closely related to the mitochondria, but we don't have them. Oh, sorry. Importantly, this one and the next one we're going to talk about both have their own DNA, which is pretty rad, actually. So mitochondria have their own DNA, which means that their replication actually occurs outside the replication of the cell cycle. There's a point in the cell cycle where we take all of our DNA and we replicate it so that we can make two cells that have exactly the same DNA in it. But mitochondria have their own DNA, and so they're not going to be replicating themselves at the same rate that our cells replicate themselves. What that suggests is a coevolutionary process, where cells haven't always had mitochondria, but rather mitochondria are intruders at some ancient point in our cells a long time ago. So the enzymes that are embedded in that inner membrane are really manufactured by the DNA that's floating around inside that inner compartment of the mitochondria. Okay, so this is the organelle that I was alluding to. It's the chloroplast. We don't have them, right? Chloroplasts are only in plants. And chloroplasts are also responsible for manipulating energy. But here, chloroplasts specifically take a physical type of energy, a photon, a light energy, and they convert it into chemical energy, specifically sugar. And we're going to talk about that process much more than you're going to want to in a few weeks. So chloroplasts. The function is to convert solar energy into sugar. You know, it's a little bit funny because if you look at how efficient chloroplasts are at actually converting solar energy into sugar, they're terrible. They convert like 2% of the available sunlight into sugar. But it doesn't matter that they're incredibly inefficient, right? Because sunlight's free. They can be as inefficient as they want. It's an abundant resource. Okay, so through this process of photosynthesis, they're able to take solar energy and really use solar energy to drive the production of sugars. Their structure, they consist of at least 3 layers of membrane. So the layers of membrane look something like this. So I'm going to and right here, this is going to be our chloroplast. So chloroplasts have an outer membrane. It looks something like that. And then they have an inner membrane that is equally smooth. So this is going to be the simplest of our chloroplasts. It's going to consist of just 3 membranes. So an outer and inner membrane, and then a series of membranes called the thylakoid. And the thylakoid looks like this. Like that. And then think about that everywhere. But they're all connected. So this little thylakoid would be connected to a thylakoid membrane over here. Like that. And this one would be connected to another one. You guys get the idea. So that's the thylakoid membrane, and each of these stacks is called a granum, or plural grana. So thylakoid, you can think about it like stacks of coins, really, that are all contiguous on the inside. Each one of those stacks is the thylakoid.